Welcome back, gentlemen. We are going to finish up this unit by doing a short lecture on mutations, which are changes in the DNA sequence. Any change in that nucleotide sequence is a mutation. They can involve something as small as a single base pair, so just that one single letter, A, T, C, or G, or it can actually involve the entire chromosome, which of course is a much larger scale because the chromosome is made up of hundreds to thousands of base pairs. Mutations can occur in somatic cells. So if you remember, somatic cells are body cells, basically anything except the sex cells. That can cause things like cancer, or germ cells, which give rise to the gametes, the sex cells. And those kinds of mutations that affect the germ cells will actually be able to be passed on to future generations. So we're going to start with chromosome mutations. We have deletions. So basically, if you take a look, this is a pair of homologous chromosomes, which means those chromosomes should be the same size, the same shape, and have the same genes. A deletion is just when a small piece of one of those chromosomes is cut off. So what does that mean? If a piece is missing, well, you might be missing some vital genes, because that's one small segment of a chromosome. Could be, it could be one gene, it could be two genes, it could be 1,000 genes. It just depends on the length of the piece that was cut off. An inversion happens, it's exactly what it sounds like. If something is inverted, it's flipped backwards. So what happens is that a piece of a chromosome will break off and will rotate 180 degrees so that now it's backwards. So imagine if you're like writing a sentence, and in the middle of that sentence, you take out a piece and you switch those letters around. The sentence isn't going to make sense anymore, right? So if you reverse all of the sequences, all of the nucleotides in that particular segment, well, now they're not going to code for the same genes. The DNA is going to be backwards, which means the RNA is going to be backwards, which means now you're coding for completely different amino acids. And then we have a translocation. So here we have two pairs of chromosomes, two pairs of homologous chromosomes. A translocation is what it sounds like. Trans refers to across, and location is, the spot. So this is when a piece breaks off of one chromosome and attaches itself to a non-homologous chromosome. So we know that during meiosis, or hopefully you remember during meiosis, oftentimes homologous chromosomes will switch segments. Sister chromatids might switch segments, which gives them, it's called crossing over, gives it greater genetic variation. Here, it's a piece of a non-homologous chromosome that's breaking off and attaching to something else. So what you have now is you have one chromosome that's missing a segment, and you have a segment that's extra on this other chromosome. Gene mutations. These are the ones we're going to focus more on. So a gene mutation is, it could be, like I said before, it could be as small as a single nucleotide, or it could be a large segment, but we're not talking about the entire chromosome. We're just talking about it on the base pair level, those A's, T's, C's, and G's, and what happens. Okay? A point mutation, to start with, a point is what it sounds like. It's a single point. So what that can mean is you're talking about a single base pair, a single nucleotide, either A, T, C, or G, and it could be the substitution, meaning let's say let's take a look at this mutation over here. So if the DNA is supposed to read CTT, but then there's a point mutation, and now you have an A where that first T was supposed to be, well, that's going to change the amino acid, because now you have a change in the DNA. You're supposed to have, in the mRNA, you're supposed to have GAA, which codes for glutamic acid. If you change that first T to an A, now your mRNA during transcription is going to read GUA, which is going to code for valine. So you've changed one amino acid in the sequence. Now, just changing that one amino acid can make that particular protein faulty, means it's not going to work so well anymore. A substitution, a single substituted letter, so if you switch one letter out, which will then change that one single amino acid, it is called a missense mutation. So it's one amino acid is affected. The rest of the protein is exactly the way it's supposed to be. Nothing has changed about the order of them. It's just that there's one amino acid that's now been substituted for another, which means the protein is not going to work exactly the way that it's supposed to. Insertion or deletion of a single nucleotide is going to cause something else that I'm going to talk about in the next slide. But basically, if you put a letter in or take a letter out, that's going to cause everything that comes after that change to also change. Okay, so that's going to be something different than a substitution or a missense mutation. A silent mutation could be as a result of a substitution, but what that means is when you change out a letter, it doesn't change the actual amino acid. So let's say you were starting with let's say you're starting with this DNA sequence CTT, and your mRNA is supposed to say GAA, okay? Let's say now this T right here becomes a C. So now you have CTC, and your mRNA is going to be GAG. Well, guess what? GAG codes for exactly the same thing as GAA. So even though you've had a substitution, it doesn't actually change the amino acids at all. So that's what we call a silent mutation. Yes, there is a change in the DNA sequence, so that's why it's a mutation, but it didn't cause a problem. The protein is still going to be made exactly the same as it was before. A frame shift mutation is what I was just talking about before when you have an insertion or deletion. If you take away a letter in the DNA sequence, or if you add a letter into the DNA sequence, everything behind that change, everything that comes after that change is going to shift one way or another, okay? So again, here's our normal DNA sequence, okay? You can read all the way through here, ACG, so on and so forth. Here's the mRNA that it codes for, and then the 3 amino acids that are coded for by those 3 codons, okay? A frame shift mutation, so now you can see the dotted box around this G. What happens if there's a deletion and now that G is no longer there, okay? This C moves up to take the place of the G, which means now instead of the mRNA reading UGC, it reads UGG. This T comes up to take the place of the C. This C comes up, so on and so forth. They all have to move up. So now the entire mRNA sequence is different after that one change. So now instead of getting cysteine, glutamic acid, and glutamine, you get tryptophan, serine, and arginine. So you have 3 completely different amino acids. So it's a completely different sequence, which means the protein that you're supposed to be making isn't going to be made at all. Whether it's faulty or not, that particular protein is not going to be made. Okay? There's one other type of mutation that I want you to write down, and it is a nonsense mutation. What that means is that at some point, a substitution, a base pair substitution, changes an amino acid, changes the codon to one of your stop codons. So instead of getting an amino acid, so it's called an early termination, so you're supposed to have many amino acids coming after that, but if you somehow get a mutation that changes a codon to those 3 stop codons, the UAA, UAG, or UGA, translation is going to stop at that point, and it's not going to pick back up again until it finds another start codon. So a nonsense mutation means you're only getting a small portion. Yes, the part of the portion that's made is correct, but because it's going to be a much smaller segment, again, that protein is not going to function properly.